hearing, smell, and taste. The pathways, the physiological pathways, okay? So hearing, this one part of this whole chapter, hearing has one cranial nerve that is called vestibulocochlear cranial nerve, okay? So vestibular means extra, by the way. I think it's extra. So vestibular means extra, and this actually vestibular part of this cranial nerve deals with balance of your body, okay? If you feel dizzy all of the time, you have problems here. And cochlear part, cochlear, the word meaning of cochlear means snail shell, because inside the internal ear, it's the shape of this part is cochlear, so snail shell. So this is for the hearing part. Cool. So hearing, this cochlea, hearing is comprised of 3 parts plus 2 membranes plus a spiral ganglia. I told you ganglia means axons. They make some sort of nerve. In vision, they were making these axons, they were going to LMN, lateral, not meniscus, sorry, LGB, lateral geniculate body, right? Here the ganglions will also make some axons. I will tell you what. So cochlea, the part which is responsible for hearing, has 3 parts and 2 membranes and 1 spiral ganglia. These 3 parts are scala vestibuli, scala tympani, scala media. Okay, scala tympani and scala vestibuli, they make something that is perilymph, okay? Lymph, which is perilymph. Perilymph is like, it's not endolymph. Actually, the name of the perilymph is done because endolymph was named earlier, and then they found another lymph, another solution. Lymph is just water. What is lymph, by the way? Lymph is water like. The meaning of lymph is water like. So perilymph is anything that is not endolymph in ear is called perilymph. That is the concept. So perilymph is majority, it has sodium ions in it, okay? It's positively charged. What about endolymph? Endolymph is made from scala media, okay? And this scala media have something called as hair cells. I will tell you what are they. So this endolymph has majority of potassium ions, and this is why the potential of this endolymph is plus 80 millivolt. It's positively charged. So it means the flow of the current is decided by the flow of endolymph. Why nature has given us endolymph? Because nature wanted the ear to have some sort of flow of current, right? We wanted electrochemical gradient. That's why we have endolymph. Cool. So this is normal. This is electrically charged. Cool. This has positive ions in it, endolymph, and it's made by media. Media means middle here. Here, media, vestibule on the top, tympani on the base. Why tympani is on the base? Because it's a drum. The word meaning of tympani means drums. So that's why tympani is on the base. Then we have media, and then we have vestibuli, the outermost, extra. I told you vestibuli means extra. Cool. What else? So this, we have other 2 membranes called basilar and resnier. The word resnier, it came from a scientist, and that's why we named it resnier. He discovered this. So resnier membrane, it maintains the flow of endolymph and perilymph. This is the function of resnier membrane. How endolymph? Scala media and scala tympani and scala vestibula. Cool? Okay. Basilar has 2 parts. Basilar means the base. So the membrane found base, it's called basilar. The membrane that is maintaining the flow of these 2 lymph is resnier. Cool. Spiral ganglia. They make axons that make something which is called as cochlear branch of 8th nerve. Cochlear branch of vestibulocochlear nerve. It's a nerve, but it's a cranial nerve, right? Okay, they make cochlear branch of 8th cranial nerve. Now this 8th cranial nerve is responsible for hearing, and the hearing receptors are located at organ of corti, right? These receptors are present at organ of corti, right? And these receptors for hearing are hair cells. Okay, the name of the receptors that are present inside the organ of corti are named hair cells. Why? Because they are thin as hair. They're like hair, and the movement of this hair will decide the potential of the cell. I'll tell you what happens. So this organ of senses sound relay information to spinal ganglion. Ganglion is just gang, a collection of cell bodies of neurons. That is ganglion. That's the definition of ganglion. Okay, so hair cells are the receptors for hearing. Okay, so hair cells can be on outer, on inside. If they are present outer, we call them outer hair cells. If they are present inside, they call them inner hair cells. Cool. Both of them are covered by tectorial membrane. Tectorial means tectus. It means it means surface. So surfacial membrane, tectorial membrane covers both of them. Okay, we know them. What about Dieter cell? Okay, before Dieter cell, let's try to understand something else. Let's look at this example. This is a OHC, outer hair cell. This is IHC, inner hair cell. They are present outside on a periphery. They are present inside. Okay, and these two cells are connected via Dieter cell. The name Dieter comes from a scientist as well. It's an eponym, Dieter cell. They just connect. They support both of IHC and OHC. This is Dieter, D. Okay, so this is Dieter cell. Let's study about outer hair cell first, and then we'll study about inner hair cell. So outer, they are present outside. So it means they work as receptors for what? For your sound, right? These outer hair cells are more in number than inner hair cell. Why? Because they are effectors. They produce effects inside the cell. They amplify and fine-tune the signals from outside. That's why they are more in numbers. What about inner hair cells? Why they are less in numbers, and more innervated. Innervated. What does innervated mean? Innervated means neurons are connected to this cell. That's it. So 90%, 95% of neurons are connected to inner cells, by the way. So this is like outside mic. This is the inside working. Cool. They are more innervated from neurons because they carry sensory information, and these are sensory receptors that convert fluid motion of what fluid? Endolymph fluid motion into signals that can be read by your brain. That's the concept of inner hair cells. Okay, we know inner hair cells and outer hair cells. Let's study a little bit more. So outer hair cells have two things: a big cilia and a small, not cilia, it's a microvilli. What is it? Well, a big cilia, we call that tallest cilia, we call this kinocilium. The name of this is kinocilium. It is a true cilia. It is a 9+2, structurally it is a 9+2 microtubule. Micro means small, tubule means they are big. If it was microfilament, it means they are really small. Tubules mean big. They are big in size. Cool. This is the same cilia that is used for movement. This is the same arrangement that is used for your movement of your eggs inside uterus, by the way. So true cilia, the arrangement is 9+2. They can change the polarity of cell. I will tell you how. This is a strong cilia. It's a big cilia. It moves. Okay. So in adult, this cochlea. What is cochlea? This organ of corti, right? This cochlea only OHC outer. This cochlea loses their OHC, the outer hair cells, outer kinocilium, because the outer kinocilium evolve into special organized organs, and this special organized organ it overdrives the function of kinocilium. So kinocilium is no longer needed. No longer needed. That's why adult cochlea loses OHC, outer hair cells. That's the concept, okay? What else is the concept? More in numbers, we know why they are more in numbers. Cool. Let's study something which is called as basilar membrane. I told you we have two membrane, resinar and basilar. Basilar membrane has two parts, I told you. What are the parts? Base, a basilar membrane has a base and an apex. Okay, base is broad here. Sorry, I'll tell you, wait. Basilar is broad and it close to oval window. I will tell you why it matters. So this is an oval window. Just imagine this is an oval window. This is the base. So base is narrow in comparison with the oval window, okay? When I say broad, it's relative. So it's relative. So it is smaller than oval. So oval window of your ear is bigger than base, and that's why the sound of higher frequency gets trapped inside, and that's how you can identify high frequency waves. We need basilar membrane base to recognize high frequency wave. What about apex of your basilar membrane? Apex is narrow in shape because it's apex. Apex means a point, a top, right? Apex predator. We are top. Apex is close to helicotrema. What is helicotrema? What is helico? Helico comes from helix, which means spiral, like this. This is spiral. Helix, DNA is also in helix shape, by the way. So helix, spiral. Trema means hole. So this hole is present where two structures they meet. Which two structures? Tibuli and tympani. Okay, how? I will tell you. This is called theory of hearing, place theory of hearing. And what happens is that scala vestibuli, scala tympani are connected via helicotrema. And this helicotrema is a pressure valve, release valve, okay, between this. And this is the reason this is the pressure valve between these two layers, and this can modulate the pressure of what? Of your sound waves. And this is the reason why you can hear low frequency waves. Okay, this is the reason. This is how you understand low frequency waves. Okay, we understand this. By the way, basilar membrane is also called frequency analyzer because one part is analyzing high frequency waves, other part is analyzing low frequency waves. We call this traveling wave theory. Same sound goes to the same sound, by the way. The same sound goes to base and then it goes to apex. And then both of them, they analyze the frequency of. Base can recognize high frequency and apex can recognize low frequency because it has a helicotrema. Okay. Let's study the concept of hair cell movement. How these receptors work. The concept of hair cell movement in response to sound. When sound hits these hair cell, what happens? First of all, let's see my description. When sound hits the basilar membrane, this membrane, there are vibration, right? Vibration, and these vibration result in motion in tectorial membrane, which is present on the top of organ of corti. If you know the anatomy of ear, you will understand it very easily. The vibration causes the motion of tectorial membrane, which is present on the organ of corti, and then this vibration, this movement causes the bends in OHC, outer hair cell. And then this bending opens up the mechanical gate for this channel. And this channel allows the movement of endolymph inside. potassiumly rich endolymph, and it changes the potential of this cell. This is how the movement of endolymph causes action potential. Okay, in response to the sound, if the movement is towards kinocilium, towards means something is coming towards you, you are gaining something, so it means it's positively charged. If it's positive, it means there is depolarization, means positive. The charge RMP is jumping from negative to positive because there is influx of potassium ions via endolymph. K influx via endolymph, depolarization, conducts nerve influx. If it's away, it means you're losing something. You're giving away your money, you're giving away your health. It's away, it means it's negative. If it's negative, it means your cell is going hyperpolarization. If RMP was minus 60, it goes even deeper, minus 70, and then minus 90. Eventually, your cell becomes hyperpolarized. More number of positive ions will be needed to depolarize the cell again. So it inhibits the nerve conduction as cells are becoming negatively charged, depressed. This is the concept. This is how I remember. So usually in other cells, depolarization is done via the influx of potassium cells, okay? But in hair cells, it's done via the influx of Usually depolarization is done with the influx of sodium cells, sodium ions. But in hair cells, the influx of potassium depolarizes the cell, the nerve impulse is generated, the action potential is generated. And then this nerve impulse goes to the ear, and then we can measure this potassium current. And then we call this thing called as autoacoustic emission, OAE, right? And there was a guy named Kemp, and the name of the guy, and he found this thing, okay? And he found this, and he named himself Kemp waves. Named these waves as Kemp waves. K current can be measured in an OH, autoacoustic OHC. This O is outside hair cells, okay? So this current can be measured in OHC, and this is a sensitivity hearing test, which is done by Kemp waves. Okay. Prone to damage. What is it? OHC, outer hair cells, are more prone to damage. Why? Why they are more prone to damage? Well, they are more prone to damage because they are metabolically more active. If something, so I was saying why OHC are more prone to damage, because OHC are present on outside, by the way, okay, and these are most damaged by aminoglycosides. These are just class of antibodies, okay, and this, for example, one aminoglycoside is gentamicin. What happens? These OHCs are really metabolically active, so they need more nutrients, right? The uptake of nutrients is higher, so higher uptake means more hungry, right? So they take a lot of things, so they are first to be exposed to drugs, any sort of drugs. So this gentamicin goes inside OHC, and then this gentamicin releases something called as free radical, H plus protons, and then this induces cell death of what? Of OHC. OHC can change in length, of course, mechanically they change in length. We call this electromotility. And this is the reason why they are metabolically more active. Whereas on the other side, these cells inside of IHC, interior hair cell or inside hair cell or inner hair cell, these cannot change their length. So they are metabolically less active. So the uptake of new drugs is less. So that's why they are the least to get damaged. This is the concept why OHC get more damaged. Okay, so this is done. Let's study the neural pathway. The neural pathway, how the sound travels. Well, the sound travels with an acronym called This well, the acronym is E. E. coli ma. E. coli ma. But I made it your ma. It means your mom has E. coli. It is a nice way to remember this because E. coli ma, I think I will forget this. But your ma has E. coli, I will never forget. Your mom has E. coli. E. coli is an infection which happens in, it's a ST, it's a urinary tract infection. So E. coli. Your mom has E. coli. What? This is amazing. I'm shocked to hear this. I'm shocked to hear this because it's a hearing acronym. Your ma has E. coli. The pathway itself, let's see. E stands for eighth nerve. Eighth nerve comes from cochlear hair cells, right? We just study about all this thing was about hair cells. And this eighth nerve is coming from spiral ganglions. Okay. So, eighth nerve goes to cochlear nuclei. This cochlear nuclei is present in the brain stem. Pons, medulla, this one. This brain stem. And then this brainstem, this cochlear nuclei goes to superior olivary nucleus. Sun. Superior olivary nucleus. Okay, they are named olivary because they were shaped like olives. The neurochemist, the neurodoctor was like, neurologist was like, okay, they look like olive, let's name them superior olivary nucleus. And after this, the impulse go to lateral lemniscus, LL. This is the main ascending tract. It goes to cortex. And then after this, the impulse goes to inferior colliculus. E, eighth, C, cochlear nuclei, sun, lateral lemniscus, inferior lemniscus. Then we have something called a medial geniculate body. We had lateral geniculate body in visual, but we have medial geniculate body in music, in hearing. Okay, so this medial geniculate body is a gateway to cortex, and then auditory cortex is reached. Okay, cool. If you have any sort of lesion or any sort of disease in these nucleus, sun, and inferior collicus, you will have problems with sense of direction. Okay, the sense of direction, right? And these two, nucleuses, they actually result in startle reflex. Whenever you get startled, scared by some sound, right? Something happens, someone claps, and you get startled, you get shocked, like something happened. You move towards it, and you want to see what happened, and you try to understand the location of the sound waves. If you don't have these two working nicely, you will not be able to have this startle nucleus and location of this sound waves. Okay, now the impulse have finally reached auditory cortex. This auditory cortex is where? It's here in Brodmann's area. Area number 41, area number 42. Area number 17, 18, and 19 were for visual. Area number 41, 42 is for music. Area number 41, what it does? It organizes the sound in notes, like piano, mapped on cortex. So, and area number 42 adds these notes, converts these notes into melodies, and we get the meaning of the sound, right? So the left hemisphere is really good at language processing, and the right hemisphere is good at music. So whenever someone is really good at music and they really suck at language, for example, someone, so they have right hemisphere really nice working. And if someone has really sucky language, they will have problems with left hemisphere. For example, an older form of piano had 83 keys, and that's how I remember area number 40, 41, and 42, because piano has 83 keys. Well, piano has more than 89 or 87, I think. But older piano models had 83 keys, so 83 equals to 41 plus 42. So this is how I remember Brodmann's area. They are also called superior temporal gyrus. Cool. They're also called hazel gyrus and planum temporal. These are just other names of it. What about stapedial reflex? Well, whenever you hear a loud noise, like a clap, a thunderclap, you automatically, this is something called a stapede. Stapede is the bone, right? Here, steps. So what happens is that stapedial reflex happens, and this stapedial reflex results in contraction of stapedius, right? And this reflex happens is like the sound that was 160 decibel lowers down to 120 decibels so that our tympanic membrane is not damaged. We need this sort of thing, okay? What happens is that 40 decibels are attenuated, right? So we also call this attenuation reflex or preventive reflex, right? If 160 decibel is, you're listening to 160 decibel, which is a very loud noise, there will be rupture of tympanic membrane. So we need stapedial reflex. What is the pathway? Loud noise, 160. Via 8th cranial nerve, this is the afferent, this is the center, and efferent. Simple. Via afferent, 8th, sun, you know this for the direction of the sound, sun, superior olivary nucleus. Via 7th cranial nerve, 8th, 7th, which is the facial nerve, goes to stapedius. And then contraction of the stapedius happen. And then attenuates the sound. Minus of 40 decibels. It prevents the rupture of tympanic membrane and your ears are fine. This is so important because if your 7th cranial nerve, the efferent nerve, is damaged in an accident, the facial nerve is paralyzed by some stroke or accident, your stapedius does not contract and it will result in hyperacusia, hyperacusis. Loud noise perceived even louder. So you don't have to shout at this person. It will be just, you have to speak with this person and he will think you're shouting at them. So this reflex can be inhibited by cannabis. So when you smoke some sort of cannabis, when you smoke THC majority and CBD less, when you smoke cannabis, the reflex gets inhibited. And that's why the volume of the music increases when you're high under cannabis. So this was your hearing part of this chapter.